I'm an infectious disease data scientist, which means that in practice I should be spending all my time sort of chasing pathogens and understanding why we get them or what the vulnerabilities are and so forth. But in reality, it means that I spend a lot of time juggling data and futzing with computers and so forth. I was on the bioinformatic team that handled the 2009 swine flu outbreak, the global pandemic. Okay, we were also the people who got the first sequences of MERS-CoV, the Middle Eastern Respiratory Syndrome coronavirus that is still blitzing through Saudi Arabia. And I was also involved in the accidental outbreak of foot and mouth disease a couple of years before that. And my friends described my Twitter feed as a constant torrent of sorrow and misery. Okay, so here are some contact details in there. I used to work for the Health Protection Agency UK, but I'm currently based at Imperial College at the historic Paddington campus. Tour buses go by underneath my window, which you can see in the upper right there, just a slither of it, and they announce the site as saying, "Here is where Prince George was born." Nothing about the discovery of penicillin that took place in the windows on the left of that picture, but there you have it. Okay. Now, I feel like a bit of a fraud today because there's not going to be a single bit of maths or a symbol or anything like that, and I don't actually even think I have an explicit line of code or graph or anything like that. What this is a war story from a research project in progress. What we did in a data-intensive biomedical project, why we did almost everything wrong, and how we managed to survive that nonetheless. So there's lots of high-level details, tools and techniques. Here's what we did, here's the things we used, here's the possible pitfalls. Not everything I talk about is Python, but Python is the glue that holds it all together, and that made it possible. So that's a good enough excuse there. Now, there are some specific names and details that I will not be mentioning because I just don't want to get bogged down into the mire of why someone's favorite database is in fact a terrible piece of rubbish. Okay, but we'll get to them as we go along. So our background is this huge infectious disease research project. Okay, the high-level concept is if a child is brought into an emergency room with an apparent fever, what have they got? Why have they got that? Why are they suffering from that much worse than another child that is not? So we're looking at the sort of determinants of disease vulnerability in there. And as a consequence, we have this vast body of data that we've built up. We are collecting information from over 20 sites, from over a dozen countries scattered across Europe and one in Africa. We currently have more than 5,000 patients and are aiming to hit more than 10,000 by the end of the year. And they have had the full diagnostic detail gauntlet thrown at them. We've got the clinical signs that doctors collect at the emergency room. They've taken their heartbeat, they've taken their blood, they've cultured their blood. Okay, we've got the timelines of everything that's happened to them. Okay, we've got all the operations they've had, all the treatments that have been given to them. We've, of course, this being a genetic project, we've gone wholly into the sequencing. Okay, we've done gene expression, all of that stuff. So naturally, this is a very huge, complex data set, and it was, of course, put into a very robust, very well-designed and intricate database. No. Okay. And thankfully, this all took place before I came on board. Okay. A few words about the world of clinical research databases. Now, there is this huge market out there for these large epidemiological trials, okay, to manage all these sort of patient records. Pharmaceutical companies use them, okay, medicos use them to run these tests and so forth, and they are very expensive, high-end pieces of equipment. I won't mention the particular one we were using, but it rhymes with Moracle. Now, of course, The design and data analysis details and so forth should have been given huge consideration at the start of this project. But as is often the case, there was this idea, we'll just buy the top of the market and everything will be fine. And that did not take place. We invested, I say we in the royal sense, of course, it was not me. We invested in this very expensive clinical research database. We invested in a third-party team to host and take care of that database, design it for us, although no one actually took care as to whether they had any experience in doing this with an epidemiological trial of this size. In fact, no one actually had experience in this database. I did from a previous job, and it is widely reputed to be a write-only database. You can record all your information in it, and it will be nice and safe in there and will never be coming out. Okay. Of course, we want to know things about this data. Okay, we want to report on it and analyze it and so forth. And there are these very intricate, complicated reporting tools in there that sometimes give you the wrong answer. For example, one time we wanted to see what sort of blood pressures our patients had, and it's giving us an average figure of 300 millimeters of mercury, which, you know, could adequately be described by this blood gushing out of these people and hitting this roof for the emergency room. The system crashes occasionally. Okay, wonderful. We can't get a direct SQL connection. That's just not possible. Okay, we can't get a stats package connection to it. Ditto. It's just beyond the skill level of most of our users to actually do anything with this thing. And so we have this huge body of information in there that we can't actually use. So what do? The whole of the project is kind of contingent upon all the information that is trapped in this system. But we do have one window, one crevice of a way of getting at that data, and we were able to convince them to send us a regular dump of the database. Now, it's not an SQL dump. It is a set of nearly 200 raw text files just poured out from the system. And so we immediately started to study them. The schema of the database is interesting, to say the least. It's changed several times during production because no one quite adequately thought about the sort of things that we might want to ask and so forth. And so several times during the trial, people had asked for features to be added, and so the schema was modified. And indeed, sometimes just entire tables were duplicated and changed slightly. So we actually had patient data lost, isolated within this system. Okay. The namings and nature of things was a unique combination between computer-generated arbitrary alphanumeric digits and operators that could not spell. There was one particular delightful part in the database where we recorded whether we took a sample of someone's blood, and it was how many of those samples we took and what the volume of those samples were. And everywhere in the database, they were systematically put in the other column. The number was in the volume was in the number. Okay? So we've got 200 files here of stuff. And I have a team of clinicians breathing down my neck saying, get us this information, it's important. I don't understand what's so difficult about it. Now, of course, when you're faced with this, we had several false attempts at trying to get things out of there. Your initial inclination, of course, is to just start writing code and we'll extract something line by line by line. But the maths of this is fairly, if you'll sit down and think about, if you spend 10 minutes working out which each field is, and you've got more than 2,000 of these fields, you're going to be here for the next 2.5 months, having worked through weekends, having worked long nights and everything, just doing this extraction, and we do not have this time. Okay? So we have to mobilize this data into something that we can actually use. And fortunately, there is this lovely tool called CSV to SQL. Okay? and it does exactly what it says on the tin. It can turn a comma-separated value file into SQL, and you can inject that into a database, and boom, you've got a database there. It's part of this, it's a small part of this lovely kit called csvkit, of course, that it has that Unix Swiss Army knife variety. I only do one thing, and I do it well, and that's work with CSV files. I search them, I split them, I'll turn them into all sorts of things. And so we were able to get these CSV files, and I can turn that into an SQL-like database. Now, you can have it automatically infer a schema, and it's pretty good at doing that. However, the date formats within the database were eccentric and differed from column to column. Okay? I could have provided it with an explicit schema, but instead I just said, let's keep it as it is and just convert it all as text. So we have here a recreation of what our original device is. Right, good old SQL. We can actually query this thing and get stuff out of there. And there we have our 200 tables. So, rather than write code line by line, how can we somehow sanitize this, clean this up, and get it mobilized in as fast a way as possible, this massive task? Rather than express it all just as code, what I did was embed the entire process as a series of tables of rules, okay? What column and table are we extracting the data from? Where is that going to? Okay? And then set up a chain of sanitization rules that goes after that. Now, the value in this is that rather than we create a large monolithic program that defies any adaptation and explanation for later modification, and we are going to have to modify this when we understand what we've done wrong, we have all our transformations captured in one place. That table itself can form the basis for documentation. We need merely write a script to process over that, and it can tell us where everything is going from, where everything is going to, and we can give it to someone and say, check this out, this is what's happening, what's wrong with that? We also this allows us to explicitly do it as an evolutionary approach because the clinicians are wanting their data now. Okay? And once again, we're not going to get it right, so we have to accept the fact that errors will be found, and we've actually recruited the users, therefore, in spotting the errors. They go back to us, we can correct that table and shoot it through. So, we get our original data in our set of rules, we run it through our sanitizers, and we have this huge list of sanitizers we've borrowed from various places. You know, this sort of philosophy that I've taken from Ian Bicking, okay? Have a chain of sanitizers to do things to think. Fix the date on this, throw it to the next sanitizer. Fix something there, throw it to the next one. If we have an error condition or something like that, error out, I can catch it, I can see what case is not being handled, and I can go back to it. Now, because we have a large amount of data recorded in different places for the same result, let's go over that again. Let's say we have the same piece of information recorded in multiple places in the database because of the changing in the schema through time. We have to, in a lot of cases, write to the same location. So in all of these cases, what we do is we run over location in the database. If it does not produce anything substantial, we don't record that information because something else might write over it later on. We then can write this to our records. We can run a post-validation process over it to check that we've actually done our validation correctly. So, for example, check logical things like the patient was admitted to the hospital before they actually left the hospital. The patient's blood pressure is actually within reasonable parameters and so forth. And we can do certain post-processing to make our information more useful for our end users. Okay? And then we upload this, or rather we create a neutral format We can transform this into a database for our users. They can criticize us, say, what the hell is this? This makes absolutely no sense whatsoever. And we can go back and we just can keep iterating over this. So we've recruited a lot of eyes to actually spot the problems in the data. Now, creating a database. Okay? This, of course, is something we're doing all the time. But as someone in epi-informatics, bioinformatics, I would be perfectly happy never to create another database ever again. And that is because they are very often created lightly for a perceived need in science and then never used. Back at the Health Protection Agency, we at one point did a census of all the databases that we had, all the ones that everyone had said, it would be really great if we had a database on. And we had nearly 100 databases we had created over the years for all sorts of things, here, there, and everywhere. The number of ones that were actually used was something like 20. And each one of those, everyone said, it'd be a great idea if we had this. So how can we shortcut this? How can we fulfill my ambition to never create another database ever again? And it has to be user-friendly to satisfy our clinicians. Okay? It has to be secure because we are talking about personal identifiable information here. There are very strict penalties if we manage to leak any medical information out there. And it has to have all the reporting tools and everything that weren't present in this high-end clinical database out there. And the answer is this thing called REDCap. Okay? Now, this is not a Python tool, but you can work with it with Python, and it is absolutely wonderful. Okay? Okay. REDCap is basically your standard let's throw a database up on the web application. Okay? It has this very easy, very nice to create, lightweight process to create databases within it. It allows you to run reports, export in a variety of formats, Stata, R, CSV, Excel, so on and so forth. It's got a REST API, so you can actually interact with a thing. You can do file sharings, stuff like that. Now, it does have slightly weird licensing conditions in that it is free, but not freely available. It's issued out of Vanderbilt University. And when you contact them, they go through this ornate procedure about wanting you to make an agreement between your organisation and their organisation, and you become a local REDCap contact, and it's all just a little bit like a cult. Okay? But in terms of being able to just freely throw a database, like a table, up on the web, it is awesome. And in fact, in a number of labs, they've started to make quite heavy use of this. Scott Burns, who wrote a couple of packages to work with REDCap, used to come from a lab where every data set they generated would just get pushed up into a REDCap installation. Okay? Tap into the REST interface, push it up, the data set is there, everyone may work with it. So that fulfills my ambition there. Now, it also has another aspect that's very useful in terms of this current situation, which is how you manage to create the databases as well. Okay. It has your standard GUI visual editor where you can select a field and so on and so forth, but it also has a thing called a data dictionary, which is just simply downloads a spreadsheet, okay, with that design information in there. You edit that spreadsheet information, re-upload it, it changes the database schema for you, and it is fairly good about changing the schema and migrating the data in the appropriate way there. So you can iteratively evolve your database in an interesting way, okay? Make things the way you need them to be very fast. Now, there is a Python package, pycap, by the aforementioned Scott Burns, okay, that talks to the REST API for REDCap. And this means that we can upload data, download data, we can modify the schema of the REST, all of the things. So now we have, boom, a pipeline. We can pour our data out of that old and crappy database that all these clinicians and research nurses are still busily entering information into. Okay? We can transform it through our sanitation pipeline. We can upload it into REDCap, and there people can criticize it, okay? And we can repeat the procedure over and over again. And we can get that going from start to finish in an hour. So that's wonderful. This saves me a huge amount of work, and I can actually get to do interesting things with that data. Okay. But here is where the catch comes in. Okay. And it's particularly, well, it's a life sciences issue, I think, but it's particularly poignant in these medical trials and epidemiological trials. Epidemiological data is some of the worst data in the world. And I know everyone has bragging rights over this and says, no, my data is the worst. I defy you. Okay? Mine is much worse. Epi data is collected by humans, of course, and so therefore there will always be distortions. A doctor is having to make a judgment on a set of symptoms in a clinic. Is this child sick? Are they severely sick? And that goes the thing. You have lots of, shall we call them, social factors involved as well. In Greece, they hand out antibiotics like candy, so you've got a bunch of children dying from weird antibiotic-resistant infections. Your Albanian contact would really like to be helpful, but he hasn't got any data, so every couple of months he sends a couple of marginal cases because otherwise you might forget about him. Okay? The British hospitals tend to act as feeders or sources for the outlying regional hospitals, so you get a strange number of severe cases appearing at your major hospitals there, making it look as if you have an epidemic. But what you have done is actually just selected them out of the greater pool of the UK. So there are many possible confounding factors. And if we're looking at doing genetic correlates as well, we don't only just need the severe cases, we also need mild cases too. If we want to understand what the differences are between someone who gets very sick from disease and someone who gets mildly sick, we need that whole span. But we have this strange distorted data set with a whole ton of missing data in there because we designed this cockeyed, overly complex questionnaire that harried doctors and nurses sitting in emergency rooms just don't have the time to fill in, so there are huge gaps all over it. So we need to explore our data. As before, we had many false attempts on going through with that. It seemed natural for a start to do a bit of machine learning over it. Surely, if we get some sort of ML technique or we get scikit-learn and so forth, and we throw it at this data, it'll be able to tell us what weird distortions are there. It'll be able to give us some explanation of where all our severe cases are coming from. Unfortunately, our data set just is too weird. Okay? The primary problem is the missing data. Okay? There is missing data from almost every record that we have within the database there, and it is difficult, if not impossible, to know how to impute or infer that missing data. Sometimes the missing data means no, sometimes the missing data just means missing. So we did some very crude, very rough and ready things on it, and if anyone has any better suggestions, I would be more than happy to hear. So we construct a severity score from our patients, an artificial number running from 0 to 5, okay, just based on their outcome. If they died, well, that's very severe, okay? If they ended up in intensive care, well, that's the next grade down, and so on and so forth. So we have this slightly arbitrary score there. We one-hot encode all our categoricals, and Lord, do we have a lot of categoricals in that data set. The countries, the particular types of ventilation used, so on and so forth. And then using Pandas, we just do one big major correlation matrix, okay, and look for what the weird things in there are. This actually turned out to be very reassuring because it told us that the doctors, when they called something a severe case, were actually usually right. And the sort of symptoms they interpreted. But it also picked out a number of other things. Weird subcategories of patients are in there that, there's a correlation with the centre that collected those patients because they tended to see a particular type of people. There's another correlation with a particular disease because some investigator has an obsession with that disease, and whenever he hears of one, he flocks in and he adds it to the trial, and so forth. So we were able to winnow out some of our weird cases from there. Our second approach was to use this other tool called Orange. Okay, now Orange is yet another data visualization and analysis tool out there, but it's good nonetheless. It allows you to set up pipelines for manipulating data and visualizing data, and you can do this in a visual editor once again, or you can do it with Python. Either way, it's all handy. The best thing that Orange has for this particular purpose is you can give it a categorical output variable, say severity, say country, or whatever like that, and then say, run through the data set and find me the best projections, or I think they call it the most interesting projections. And it will come back, often with dozens of graphs, and say, this one looks quite interesting, and you can see how it's separated out all your patients in ways that they shouldn't have been. All our Austrian patients are shifted out and sitting separate from our English patients and our German patients, and so forth. So we're able to identify another couple of cohorts of weird things in there. Now, okay, we finally, after the end of this terrible data, after the end of kind of pick out cases that are actually cases that are worth investigating and so forth, we now actually want to get something out of this information. And disease genomics, I would propose, is one of the most interesting data analysis problems there are, because it's a classic friend of a friend problem. Right? Disease is a multi-level phenomena. Many things can go into causing you have a disease. Do you catch it easier? Are you resistant to it? Is there something within your system that means that you have a more severe case? Is it your location that you're in? And so forth. And we can get the same phenotype, the same outcome from a whole series of different changes within a person. So a complex macromolecule somewhere in the body is malfunctioning, okay? And this causes you to suffer severely from infection. It could be from something that is regulating or modifying that particular molecule. Or it could be from the piece of nucleic acid that's transcribed off that goes to create that macromolecule. Or it could be the gene that codes for that nucleic acid. Or it could be all these other genes that interact with and control these things. So we are very often talking about causes are several steps removed. And we need to examine these things within that context. We can't just say, let's have a look at all these patients and look for the common cause, because while they may have common symptoms, they may have different underlying causes due to the complex pathways underlying them. From these analyses, we also get a huge amount of noise as well too. And this is something that comes through in all these life science problems, that almost in a sense getting results is not difficult, it's picking out the interesting results. It's picking out the useful things. So, because this is data science and no one does anything as boring as a database anymore, let's go and use a graph database. But this actually makes great sense. I am a rank beginner in graph databases. Okay? And there are other people at this meeting who are much better experts at it. But to give you the high level, it would be much like our NoSQL or object databases where we're storing things in the database, except that relationships between objects are a first level citizen. Okay? Graph databases are very interested in the connections between things and how we can traverse them. So we can ask all these questions like, these two objects here, the things they connect to, what do they have in common? And many more complicated questions. Now, the one we've chosen is Neo4j. It is the most popular one out there, and there are various Python tools that work with them. Now, this allegation has been made that in the future, maybe all databases will be graph databases. I think that's fairly dubious. In theory, there is probably nothing here that you couldn't do with a conventional database, but it's the level of expression that's much more useful and interesting for us. So let's get all this results, all this information there, transform them. Let's connect it to a whole bunch of old genomic and clinical information and all these databases out there, and let's see what we come up with. Schema design for this sort of stuff is hard because your choices about relationships invariably impact the sort of queries that you can do later. And we had lots of false starts about how this data would be transformed. Fortunately, in terms of getting the existing corpus of genomic databases and pathway databases and all this other information out there into this pool, someone's already done the hard lifting. These guys called Bio4J. The installation is enormous. It's over 1 terabyte of data. But when you merge it all in and you mix it all up, you get something like this. And now you know you're at a data science conference because I put up the spaghetti picture at last. Yeah. And we can actually start to interrogate this data and ask it questions like, these two patients have the same outcome. What defects or variants have we seen in their genes, and how can those be linked together in the variant pathways that are out there? And we can actually start to understand the research project that we set on all the way back when. Yeah. So, a couple of take-home lessons and things like that. And they'll probably not be very surprising, things like that. Data analysis and data design really has to be done at the beginning of the project rather than during or in flight when or whatever. And this point is so very obvious, but in academia and research projects and so forth, it is so often thought of as an afterthought. Data science and analysis, in a sense, we've allowed it to be turned into mere programming. So you have a pile of data dumped on you and said, make sense of this. Whereas all of these issues, all of these months spent on trying to transform this data, trying to make sense of it, throwing out all these cases that we've carefully collected and so forth, could have been avoided if we'd started to think about these issues right back in the beginning. And this is so obvious, but it's going to happen again and again. Of course, this whole academic research project thing is very different from a lot of the things that people are working in here, but these genomic projects these days, they are all about information and data analysis. And yet at the same time, academia and research can be very hostile to these data science projects. Terry Atwood had an interesting paper recently on biological research databases, examining the half-life of them. You set up a database on some vital target, how long is it likely to hang around? Sometimes the database is shut down before the paper has been published on it. Okay. Data science is not something that gets taken very seriously in academia, but it is something that it needs. Life science data problems, the sort of ones I've talking about, are in a sense very different from the things that other people look at here. People talk about, I've got 1 million customer rows. We need to get eyeballs on our website. I always say, I've got 5,000 records and they're all terrible. Yeah. Very often, the issue is being able to make sense of large, but not vast, but very detailed records and get them out of there. Evolution beats revolution, and tools are not a lifestyle. And these are two things I feel kind of very passionate about. All too often, I've wasted huge amounts of time on tools and revolutionary techniques and things like that, that just said if I would invest much more time and do these particular approaches, then your life will become wonderful. But being able to do this iterative approach to solve this data problem, being able to get something out to people and having them criticize it wildly and then go back again and again and set this chain up, worked in this particular case. Using something as dumb as SQL and so forth worked very well. So we've got expertise in that, it just works. Redcat just works, so forth. Tools that just work are great for me because it means that I can just do these things and then get on to the actual interesting part of the work. I'm sure you can Google for all the things that I uploaded or mentioned along the way. Here are some URLs in there. And that's it. Thank you very much.
Hi. I've don't know RedCap very much, but when you were describing it, I was wondering how much help was giving you from maybe someone who knows SQL a lot, and given CSV files, you can just load directly that into tables.
Right, okay, okay, there's lots of points there. Of course, one of the issues of academic informatics is that you're very often stuck on your own, and it's like you to solve this problem and so forth. So I did not have an SQL expert there for me. A lot of the metadata and information, indeed almost all of it, had just been stripped from those tables by the time it got to me. They were really very context-free. And the internal schema was one of the most bizarre things I'd ever seen. Possibly someone who was an SQL expert might have been able to make better sense of it. I fancy not, because so much of it had been automatically generated. It was a very strange, hyper-denormalized structure inside there where you would have the same bit of information occurring 10, 12 times in different tables. Maybe someone might have been able to make better sense of it, yes. That person was not available to me.
And maybe I have a second question. I mean, it's all related, and it's probably a similar answer, because I'm coming more from a software engineering background, sure. and just in my last job, I've started working with data, which fascinated me, and didn't know even very much on having just CSV files and probably loading directly into SQL. But also I was asking myself, well, is it easier if those CSV information, which we don't know anything about the schema, putting on a schema-less database, let's say famous NoSQL. I mean, I'm not saying crap databases because that's much more complicated, right? Sure. But something like MongoDB, which is just documents of...
Okay, right. Now, we did consider a schemaless database along the way. I'd done a little bit of playing around with such things in consideration of them, and I certainly won't say that my opinion is a hugely informed one. My impression was that schemaless databases do have a schema, it's just implicit.
Right.
Okay, and so therefore, a schemaless database, in a sense, is a useless database if I don't know what the things are that they're in there. So it may have been a better solution in the long run. I don't know, but it was probably not a time to invest in risky technologies when we had this whole trial hovering on this particular outcome.
Right. Okay. Thank you.
Good.